morning, everybody. It is my great pleasure to introduce Dr. Monica Perez. I met her on the Sensory Motor Integration Review Panel, and it has been a wonderful visit having her here and just getting to know what all really, really wonderful stuff she's been doing and how much her work overlaps with some of the activities that we're doing. She actually started as a physical therapist and then did a research project at the Miami project with Adele Phil-Fatay and did her PhD there. She then went to Denmark for a postdoc with Jens Nielsen, and then to NINDS to work with Leo Cohen for another postdoc, and then she is now at the University of Pittsburgh in the Department of PM&R. She has a very impressive list of publications for being fairly early in her career, which makes me incredibly jealous. And she's got all kinds of wonderful, outstanding awards for, outstanding this, outstanding that. And so we're expecting an outstanding talk, and I'm sure she will absolutely deliver. So welcome, Monica.
So, thanks, Don, for inviting me to be here today. So, is this working? Can you hear me okay? Okay, so the work in our laboratory primarily aims to understand how the brain and the spinal cord contributes to the control of voluntary movement. And we test intact humans and also people who suffer from incomplete injuries to the spinal cord. For that purpose, we use electrophysiology in order to assess and characterize the transmission of some of those remaining or residual pathways that are still available after an injury to the spinal cord. So during my presentation today, I will give you some general information about spinal cord injury, and I will talk a little bit about some of the anatomical consequences of spinal cord injury, particularly consequences that affect the corticospinal tract, which is directly involved in voluntary activity. Then I will show you some of the new modern electrophysiological techniques that we are using in humans today in order to understand how the primary motor cortex might be involved or contributing to different types of voluntary movements. Then I will give you examples of how our group and other groups have used this electrophysiology in order to characterize changes in motor cortex and changes in corticospinal transmission in people with injuries to the spinal cord. And finally, I will give you a direct and a concrete example of how we were able to use electrophysiology to actually not only target plasticity, but to use the electrophysiology to guide the intervention that we use. And we think it might be a new avenue for future therapeutic approaches. So spinal cord injury, we know that there are over 12,000 spinal cord injuries in the United States every year. More than 300,000 individuals suffer from spinal cord injury. 81% of those spinal cord injuries occur in men and between ages of 16 and 20 years old. And the average age is around 40 years old. 51% of those individuals have a tetraplegic injury. That means, or tetraplegic, this means that their injuries affect the cervical spinal cord and they have incomplete injuries. And this is the population that we are actually targeting and trying to further understand in our laboratory. 75% of spinal cord injuries, as you well know, are a result of a trauma or a contusive injury. And the most common causes for a trauma are traffic accidents, falls, 27%, violence, 15%, sports, and many other type of accidents. So what are the anatomical consequences of spinal cord injury? So this is on the left side. You can see a staining from a human spinal cord. This is a section in the cervical region. And you can very well see the very well-defined gray matter and the intact white matter. And I'm just going to point out where in the dorsolateral aspects of the spinal cord, where the corticospinal tract is mediated, it's located. So we have a large portion that is located in the dorsolateral part of the white matter, and we also have some small portion that is in the ventral part of the spinal cord. And this is important to consider because this tract tract is largely involved in every movement that we have to perform. So this is a staining in an individual who suffered from a chronic spinal cord injury for approximately 3 years. And this is a staining. This is a cross-section from the epicenter of the injury. And you can see that this is the dorsal part of the core. This is the ventral part of the spinal cord. You can see that there is a large loss of tissue, particularly in the area that corresponds to the gray matter, where all the interneurons and all the motor neurons are located. You can also see that there is some remaining tissue, and particularly in some of the area where the corticospinal tract is located. So this subject had some residual voluntary control. And these are the pathways that we are trying to evaluate or assess using electrophysiology. We are also doing some neuroimaging in our lab. So this is an image of the cervical spinal cord, also in a healthy control. This is at the C3 level. And you can see that this is the cerebrospinal fluid. And we can have, this is the intact spinal cord. You don't see a very well resolution of the gray matter and the white matter. But here, what is important is that this is an image from a person who, I will show you later some data, some physiological data. He had an injury. This is the epicenter of the injury. This is a chronic injury. And apart from injuries being bilateral, you can also see that we can also detect some changes more pronounced in one side versus the other side of the spinal cord. So we are trying to use this imaging marker and combine that with our physiological markers in order to understand where are the deficits, and if there is recovery, what could potentially be a marker for recovery. So what are the functional consequences of spinal cord injury? So I'm going to be brief in this area, but this is just a very simplistic cartoon to remind you that when we have an injury to the spinal cord, we don't also have interruption of descending motor pathways that go to the spinal cord, but there is also a large number of ascending sensory pathways that are being affected. And that might contribute to also the deficits that our patients are being affected during movement. There is a degeneration of the distal part of the damaged axon, and there is deficits in the connection between descending axons and spinal motor neurons, which is what I will try to focus today. Can we, by using electrophysiology, access that area, that interaction in the spinal cord? We use the ASIA classification to define our patients, the level of complete or incomplete injuries to the spinal cord. And we primarily are testing individuals with ASIA-C or ASIA-D, which means they have an incomplete sensory and motor deficits after the injury. And we also test individuals who have injuries at the cervical level of the spinal cord. So, modern electrophysiology, since I received my physical therapy degree many years ago, I think that has been continuously evolved. But if we go back to the first publication, the first paper that showed that you can successfully stimulate the brain in humans, this was in 1980. So this paper was published in the Journal of Nature, and Morton and Merton in the UK, they used two small electrodes over the surface of the scalp. One was positioned over the vertex, and the other one a few centimeters lateral to the vertex, over the hand representation of the motor cortex. And with very high density currents, they were able to elicit responses or evoke responses in different muscle groups. So that in the 1980s was quite revolutionary, to be able to stimulate motor cortex and to be able to evoke responses. The main problem, and we discussed this with some group of people last night, is that this was uncomfortable. So in the 1980s, there was a clear need for a new technique that allow us to do electrophysiology, to study corticospinal transmission, but that was not painful or non-invasive. So this is the first group. This is the first group who actually published a paper. This paper was published in Lancet in 1985. So for the first time, they were able to use magnetic stimulation over the motor cortex and to elicit motor evoked responses in upper limb and in lower limb muscles. So this is the type of stimulator that they use. This is Risa Jalinus, who is still working with the MaxStim company, and this was his PhD thesis. This is the coil that was used at that point. And this is just a reproduction of the responses that were reported in that paper. So this is what they did. They used the coil. They used a strong magnetic field, which is approximately 2 Tesla. When you position the coil over the area of the primary motor cortex, you can generate an electrical current that passes through the scalp and generate an electrical field and is able to depolarize some of the structures in the brain. So this is what you see, basically. This is electromyographic recordings from thinner muscles. So you stimulate over the motor cortex. This is the stimulus artifact. And approximately in this subject, 23 milliseconds later, you can evoke a potential. Okay? And this is coming directly from stimulating primary motor cortex and recording on the surface from EMG electrodes. If you position exactly the same coil over the peripheral nerve and use the same intensity, you can also evoke a response. It's a biphasic response, which have a very similar shape, but have a very different latency, because this is coming by just direct activation of the motor axons. Okay? So at that point, in 1985, this demonstration allows us to access the brain, the corticospinal tract, which is important because it's involved in voluntary control. But we didn't know what are the mechanisms mediating. So since 1985 until today, many groups have tried to understand what are the mechanisms and the pathways that contribute to changes in latency and also in amplitude of the response. So I'm going to just show you what I think are some of the most important publications that contribute to our understanding of neurophysiology of magnetic stimulation. So in 1996, since 1996, people were simultaneously doing not only stimulation of motor cortex using TMS, but also simultaneous recordings from the epidural space. These are from patients who had epidural electrodes implanted in the spinal cord for other purposes, primarily for pain control. And simultaneously, they have electromyographic recordings. In this case, it's the finger muscle. Okay? So if this is a cartoon showing you the position of the coil, so this is one of the coils that we use to stimulate the primary motor cortex. If you position the coil with the handle in the horizontal region, the current is flowing from lateral to medial direction. And what you see in the epidural space is a very short response that most likely through the latency, we understand that might contribute or might be elicited by direct activation of the corticospinal axons. And then later on, you can see a series of responses that happen at an interval of 1.5 milliseconds. This really largely resemble the electrophysiological recordings that have been done in primates, in cats, in animal models by stimulating pyramidal tract. So what is interesting is that when you rotate the handle, the position of the coil, so in this case, the current is going from posterior to anterior direction, you also elicit different type of descending volleys in the spinal cord. And that is important because that indicates that when we stimulate the brain, we can target different type of structures. Okay, we can perhaps target some interneurons, we can target the axon more directly. So that tells you that this could be potentially a useful tool to understand the control of movement. So when you rotate the coil and you use a very similar amount of intensity, you can elicit, you don't see the short D wave at the beginning, but you see a series of oscillations who are likely coming from indirect activation through interneurons. There's still a large amount of debate through the origin of these responses, and we're doing some work that I will not show today, but to try to understand the mechanisms also that mediate these responses. And nobody have reported these responses after spinal cord injury. There's no direct indication of how are these descending volleys organized in people who have chronic spinal cord injury. We have tried to do it in Pittsburgh, but we need to record from the epidural space. So we know that there are demyelinization, and there's loss of axons, and most likely the synchronization of these descending waves will be altered or changed after an injury, but there's no direct evidence yet. And importantly, we can record the responses on surface EMG, and the amplitude of those responses is also changed by the volleys. So this is telling you that we are activating different pathways. Later on, these experiments were combined by pharmacology, pharmacological agents, in order to understand if we could even go any further to understand mechanisms of how the brain controls movement. So since 1990, there was a series of pair stimulation protocols that has been tested. And the idea is not to only stimulate the brain with a single pulse, but use two pulses at a different intensity or at a different interval to try to understand the effect of one conditioning stimulus over motor cortex on the size of a test response elicited also over motor cortex. So I'm just going to show you one example. This is one of the paradigms that we are also widely using in humans today. And we have some evidence to suggest that perhaps GABA-B-mediated receptors, GABA-B-mediated mechanisms are involved in this mechanism, in this suppression. So the coil, again, is in this position primarily because, as I showed you before, we can target indirect or later I waves, descending I waves. So we stimulate motor cortex with a single pulse, and these are recordings coming from the first dorsal interosseous muscle. In a second condition, we can stimulate through the same coil the motor cortex, and we give one pulse at the suprathreshold intensity, so this pulse still is eliciting an evoked response. And in this case, 100 milliseconds later, we elicit a test stimulus at the same intensity. And you can see that the conditioning stimulus suppress the size of the test stimulus when we use suprathreshold intensity. If you use a pharmacological agent as a baclofen, for example, which is a GABA-B agonist, you can see that this suppression is even more pronounced. Okay? If you do epidural recording simultaneously with this type of experiment, you can also observe that at the same time that the MEP or the evoked response is suppressed, you see that the later I waves are being suppressed. So together, the evidence indicates that it's likely that this suppression in this response is coming or mediated by GABA-B mechanisms. So where are we today in 2013? So we have a variety of instruments that we can use to stimulate motor cortex. This is just some of the coils that we have in our lab. This is a figure of, this is a circular coil, a figure of 8 coil. This is a bat wind coil. This is a cone coil that we use for the brainstem or for stimulation of the cerebellum. We can stimulate motor cortex at very high frequencies, even 20 or 50 hertz. There is evidence that we can increase temporarily excitability or the size of those responses, or we can decrease the size of those responses for even up to 30 or 1 hour, 30 minutes or 1 hour. We have even more evidence now that this rapid stimulation might be able to be effective depending on which neuronal elements you're activating by the stimulation. So the large variability that has been reported in the literature since the first paper, since 2005 until today, is to some extent perhaps coming because the technique is more effective in individuals in which we could elicit some later I waves. And if you're more interested, I can show you how we can test that hypothesis. We can also combine transcranial magnetic stimulation with neuronavigation system to evoke or try to activate areas that are non-primary motor cortex. In this case, the premotor cortex. We can go to dorsal and ventral premotor cortex, supplementary motor area, and start making inferences about how the system could potentially work. So we have used this technique extensively in humans, in people who had chronic injuries to the spinal cord. So I'm going to show you some of what I think our group, but other groups have shown as some of the more classical deficits in corticospinal transmission in people with chronic injuries to the spinal cord. So the first one is that there is an impairment in the latency of corticospinal responses. Okay, so this is raw data. These are recordings from, this is the stimulus artifact. These are coming from the first dorsal interosseous muscle. So in a healthy control, if you look at the latency of this response, it's around 20, 21 milliseconds, depending on the length of the arm. If you have an individual with spinal cord injury, this is a subject with a chronic injury, the latency is much more prolonged. On average, there has been report about 5 to 10 milliseconds longer in individuals with spinal cord injury. And if you think about the anatomy, just you go through the area of the injury, which could affect 1, 2, 3, or 4 segments of the spinal cord. So this is one of the very reliable measurements, and I think most neurophysiologists will agree that this is a deficit that we can quantify. What is interesting is that this deficit is present in the acute phase of spinal cord injury, and is equally present in the chronic phase of spinal cord injury. So it's not the result of the acute injury. Okay, there are several studies who have looked at changes in latency in different phases, and this mechanism doesn't change, remain to be constant. Another deficit are changes in the amplitude of corticospinal responses. So if you look at a healthy control, if you stimulate motor cortex again, and you look at the maximum size of evoked responses with maximal stimulation intensity in the first dorsal interosseous muscle, in most people it's around 5 or 6 millivolts. If you look at a patient with spinal cord injury who has some remaining voluntary control, the amplitude is around 2, 3, or 4 milliseconds, depending on the integrity and the extent of the damage. But we can detect changes in amplitude and also changes in latency, which are quite consistent by using this technology. Another deficit that we have been able to detect is changes in threshold. We can stimulate, we need more intensity in order to elicit response, corticospinal responses after spinal cord injury. When we stimulate during voluntary activity also, the slope of the input-output excitability curve is much more reduced in people with spinal cord injury. So there's several measurements that allow us to make inferences about these deficits in corticospinal transmission. So we can also assess excitability in the motor neurons after a spinal cord injury by using electrophysiology. So we have other groups, and our group have been able to demonstrate impairment in the number of motor neurons that remain active in people with chronic paralysis. So in this case, this is raw data from a healthy control. We stimulate. These are recordings from the first dorsal interosseous. We have the tendency in the lab to look at intrinsic finger muscles. So you see the latency is much shorter because we go from the stimulating area to the muscle. We don't go back to the spinal cord. And the maximal response is around 21 millivolts in healthy control. In somebody with spinal cord injury, this is about half of the size. Okay, depending on the extent of the injury, we have patients in which we cannot even elicit a 1 millivolt response, a very, very small number of motor neurons that have survived the trauma. We could also assess the excitability of those motor neurons. So if we keep increasing the stimulus intensity, we can reactivate some of those motor neurons antidromically. Okay? And we can examine, or we can quantify the amplitude of this antidromic activation and the persistence, how many times we can reactivate those motor neurons. And you can see in a healthy control, this is the same muscle that I'm showing you here. This is the first dorsal interosseous. The responses are around 1% of the maximal motor response. if you have an individual with a spinal cord injury, the amplitude of this reactive antidromic activation from motor neurons is about 3 to 4% of the maximal motor response. So that indicates that there's motor neuronal excitability that is increased after spinal cord injury. So that is also another index that is able, physiological index, that is allow us to detect changes in the spinal cord. So we can use electrophysiology not only to detect changes in corticospinal transmission, but also to detect some reliable changes in the spinal cord, in the motor neurons. So how do we use those information? So in our laboratory, my main interest is to understand how movements are organized or reorganized after the injury and their mechanisms. So we have a series of experiments in which we look at transmission in all those pathways in the side contralateral to the movement, okay? So we stimulate the motor cortex contralateral to a limb that is performing a movement. And we test very simple tasks, isometric index task, finger task, isotonic task. We change the velocity, we change the amplitude of the movement, and then we use all these physiological markers to understand how are they modulated by movement. Since we also know that during voluntary activity in a healthy control, there's also a number of changes that occur in the contralateral side that remain at rest, we have in parallel a series of experiments where we also look at the contralateral side. So what happens if you perform a very strong voluntary contraction in one side, what happens with the resting side after spinal cord injury? And one of the hypotheses is that perhaps this will be affected because most of the injuries to the spinal cord are bilateral. You have a lot of areas within the ventral part of the spinal cord where all those commissural interneurons are located that are disrupted by the injury itself. So we look at the system not only in the movement side, but also in the contralateral resting side, and we use this information to target plasticity. So I'm going to try to give you now the second part of my presentation, and a concrete example of how do we use all these physiological measurements in one concrete example that may help to promote recovery, we think, or at least some aspects of voluntary movement in patients with spinal cord injury. So this is a recent study that was completed by Karen Bundai in the laboratory, and we look at motor recovery by strengthening residual corticospinal synapses after human spinal cord injury. So the first question that we ask is how can we induce plasticity in the corticospinal pathway? I just show you some evidence that we can assess transmission in the pathway, but how can we change excitability, or how can we make that transmission more efficient? So there are different methodologies that have been used today in humans to change excitability. We could have use repetitive transcranial magnetic stimulation over the motor cortex. You can stimulate at very high frequency and change the size of the responses. That's one possibility. You could also use electrical stimulation. You go to the peripheral nerve, or you go to the muscle itself, and you repetitively stimulate, and perhaps you can also elicit temporary changes in excitability. And actually, there are several reports that have shown that that is effective. So our approach, and we also know that the improvements are temporary, and the improvements are limited, and we need perhaps another approach in order to target the system. So if you look at animal experiments, there is quite strong evidence that long-lasting potentiation of synaptic strength can be induced by precisely timing the arrival of presynaptic action potentials prior to postsynaptic depolarizing action potential. And this process has been known for many, many years, and is known as spike timing-dependent plasticity. Okay, has been primarily shown in animal experiments, but also in humans. People have demonstrated that if you induce this phenomenon of plasticity by pairing magnetic stimulation over the motor cortex and peripheral nerve stimulation, and the bolus will arrive more or less simultaneously at the level of the motor cortex, perhaps you could potentiate excitability in the corticospinal tract. So then the next question, this seems to be a very strong mechanism. Can we use it in humans, and where do we target this mechanism? Do we target the motor cortex? Do we target the spinal cord? We can manipulate that by electrophysiology. We can manipulate the arrival of the pulses to be at the cortex, at the muscular junction, or at the motor neurons. So this is, I think, where the decision to target, where the electrophysiology becomes very important in order to guide and help us to decide where do we target the plasticity protocol. So a series of experiments that we have been conducting during the last years have suggested that the spinal cord is a critical site for modulating corticospinal transmission in humans with spinal cord injury. And I'm going to show you 2 examples only of what do I mean with the critical site and where is this physiological evidence coming from. In one study, we demonstrated that this perhaps could be the target. So this is raw data from a healthy control. This is raw data from a patient with spinal cord injury. These are responses coming from supramaximal stimulation of the ulnar nerve. So these are F waves, so index of excitability of the motor neuron, and this is the maximal motor response. So you can clearly see the F waves here. So in this task, individuals were instructed to perform a very strong voluntary contraction with one limb, and we look at the contralateral side. So you can see that during strong levels of effort in a healthy control, the F waves are present very often, and the amplitude is also larger in relationship to a resting condition. But look what happened in a patient with spinal cord injury. And this patient is able to perform very strong levels of voluntary control. Okay? So there's no deficit in force in this patient that I chose for the example. So you can see that this individual is not able to facilitate the responses coming from motor neurons. Not only the persistence is not increased, but the amplitude is not increased, okay, which was contrary to our initial hypothesis. We did a lot of electrophysiological markers, and what we found is that the responses coming from motor neurons were directly related to the amplitude of corticospinal responses. So in those individuals in which we were able to facilitate corticospinal drive, in the resting side, were individuals in whom we were able to elicit responses from the motor neurons. But less corticospinal drive was related to less activation of the motor neurons. So that was one evidence that the spinal cord, the responses there, were quite critical for facilitating transmission. Then we conducted another experiment. I'm just showing you 2 examples. But in this case, we used a pair pulse stimulation over the primary motor cortex. So this is results, raw data from a healthy control. This is a test response, a single evoked response in the first dorsal interosseous muscle. And this is a conditioning stimulus over the test response. And this is tested during voluntary activity. So during voluntary activity, you will expect that there is less inhibition because all pathways are active because you are involved in voluntary control. This is what happened in an individual who had chronic spinal cord injury who never took baclofen. So you can see that the conditioning stimulus is not able to decrease inhibition of the test pulse, but there is quite pronounced inhibition during voluntary activity. This individual, most of our subjects have clonus, and we were able to quantify the frequency of this activity, the duration of the burst, and the interval between each one of the bursts. What is interesting, we still don't know very well the mechanisms involving clonus. But this evidence suggested that individuals who have more pronounced inhibition in this technique were individuals who have more prolonged interval between the bursts of clonus. And this is, as I showed you earlier today, this mechanism might be mediated by GABA B receptors. So we are suggested that perhaps one of the mechanisms that are contributing to this pronounced clonus that we see in patients is, again, this GABA B mechanism that may be mediated by subcortical pathways. So this is the second type of evidence that suggests that inhibitory pathways that are likely mediated at the spinal cord might contribute directly to EMG activity that we can record. So based on this evidence, we decided to target the spinal cord. And we have more evidence, but for purpose of time, I'm just refer to these two experiments. So how do we induce this spike time-independent plasticity in humans? So this is just a cartoon showing you we stimulate over the hand representation of the primary motor cortex. We go to the ulnar nerve, and we use electrical stimulation over the peripheral nerve. Okay? And our goal is to arrive to be able to modulate the time when descending bolus elicited by TMS or antidromic bolus going back to the motor neurons elicited by peripheral nerve stimulation arrive at the spinal cord. And this is a very simplistic cartoon, but I'm going to show you how did we determine that. So our hypothesis was that the arrival of corticospinal bolus prior to motor neuronal discharge at residual corticospinal synapses will enhance voluntary drive. And this is based on all the animal literature and also in some human literature before. So the question is here again, how electrophysiology becomes quite relevant. How do we determine the proper time to for responses to arrive at this terminal before we depolarize the motor neuron? So it's beautiful. We can use electrophysiology again, okay? So this is a number of physiological markers that we could use, and I'm going to show you only 3 of them that allow us to make calculations of central and peripheral conduction time, and just make changes and quantify latencies, which are, as I mentioned to you before, are quite reliable. Over time. So one of the first measurements was stimulating the motor cortex with transcranial magnetic stimulation. We asked volunteers to perform voluntary activation of the muscle, and we calculate the latency. This is in a healthy control, and this is in a patient. Okay? Then we also stimulate the motor axons at 2 different levels. The first one is to activate the axons at the ventral root. So this is the latency for a healthy control, and this is the latency for spinal cord injury patient. And then we go and stimulate the motor axons more in the periphery. So this is the latency for the motor response in both populations. So by a subtraction of latencies from motor evoked responses from the periphery, and accounting from some time from these pulses going back to the spinal cord, we can make an estimation of when impulses will be at the presynaptic or at the postsynaptic terminal. So we tested 3 protocols. Okay, we tested 18 patients with chronic cervical spinal cord injury. They participated in 3 different sessions. This was a crossover design. So this was separated by at least 1 week in between the sessions. They did what we call the +2 protocol. This means that the descending bolus arrived 1 to 2 milliseconds before the antidromic bolus arrived to the motor neurons. In a second protocol, is the -15 protocol. We stimulated the motor neurons 15 milliseconds before the descending bolus arrived to the spinal cord. And this was our control protocol. We also stimulate the antidromic. The antidromic bolus arrived 5 milliseconds before we activate the descending bolus. Okay? And I can, if somebody wants more detail, I can explain you exactly why did we decide those time intervals. But we stimulated, we used 100 pair of pulses. We stimulated at a 0.1 hertz frequency for 17 minutes. We used 100% of stimulation of the motor cortex for the descending bolus, and we use supramaximal stimulation for antidromic bolus. So before I show you the results, I think what is quite important also is to examine, are we doing something different during each of these interventions? Are we driving the spinal cord in a differential manner? So we were able to record also during the intervention. So is it different? Okay. So this is raw data from one of our participants. This is the 3 different protocols. This is the F wave in a resting condition. And this is the size of the F wave when we apply simultaneously the stimulation to the peripheral nerve and to motor cortex. This is what happened in the +2 protocol, -15, and -5 protocol. And this is the group data. We stimulate 100 times. This is the dotted line here is the resting size. So you can see when we stimulate together, okay, but the impulses arrive 2 milliseconds before, in the presynaptic terminal, this is the difference in amplitude of the response. It's very dramatic, okay? So this is the average of the first 10 responses, and then we go up to 100. So this is the intervention, basically. This is the amplitude of this response when they continuously receive the stimulation. Then in the -15 protocol, this is what happened with the size of the responses. So we use the same intensity. Here, the only difference is the interval between the pulses. And you can see how dramatic is the change on the amplitude of the F wave. And then in the -5 protocol, using exactly, again, the same intensity, but just changing the time of arrival, you can see that also there is a consistent difference in the intensity. So this data suggests that to some extent we are driving the spinal cord differentially during each of the interventions. And this is the data from our patients showing you that we were also able to drive during the intervention the spinal cord differentially. So I'm just going to show you 3 results that I think suggest or support the hypothesis that we were able to elicit spike time-dependent plasticity in humans with spinal cord injury. The first one is that excitatory and inhibitory changes in motor evoked responses mimic the reversible spike time-dependent plasticity that has been documented in animal experiments. So this is raw data from a patient and from a healthy control. This is an average of 30 responses before the intervention. This is 17 minutes of stimulation, and you can see 10 and 30 minutes after stimulation, the peak-to-peak amplitude of the responses is increased. We look at the group data, and you can see that this sync occurs immediately after the intervention and is preserved for at least 30 minutes. And it follows a similar pattern in a patient with spinal cord injury. Importantly, this was specific to the +2 protocol, and the -5 protocol, we don't see the same changes, even though we stimulate for the same time and the same intensity. If we do the opposite, if we target the motor neurons before the descending bodies arrive in the presynaptic terminal, we observe that the peak-to-peak amplitude of the baseline responses is decreased for 30 minutes in a healthy control and in a patient with spinal cord injury. So you can see that those changes also were significant in both populations. So you can even upregulate or downregulate the size of responses by just changing the timing of stimulation. The second evidence is that we were able to change responses by magnetic stimulation and also by electrical stimulation. If we propose the hypothesis that this is happening in synaptic transmission, it's important to know if the origin of these changes are really subcortical. So we were able to, and we discussed this a little bit last night, so I put the slide. So we stimulate electrically primary motor cortex by using pulses between 50 and 200 microseconds and an intensity between 100 and 300 milliamps. We also went to the cervicomedullary junction, and we stimulate corticospinal axons through the brainstem. And we use similar intensities and slightly more intensities and similar pulse duration. So this is what you see. This is data from a patient. These are responses coming from motor cortex. You can see the latency of 27 milliseconds in the first dorsal interosseous. And from cervicomedullary junction, you can still see the biphasic response, but the latency is approximately 4 milliseconds later. Okay, so this response is not contaminated by any inputs from motor cortex. We are directly activating the axons. So we use 2 pair pulse protocols, and I'm just showing you part of the results here. So you can see that the +2 protocol not only increased the amplitude of responses from motor cortex, but also responses from cervicomedullary junction were increased to the same extent, and responses were suppressed when we use the -15 protocol. Okay? And the last evidence is that we were able to potentiate voluntary motor output. So this is raw data from a patient. They have to perform a fast ballistic voluntary contractions, isometric voluntary contractions with the index finger. They had control. They have a visual display on the computer. They were able to control 80% of the movement, and the last 20% of the movement was controlled by the computer. So they don't have any way to voluntarily make more or less output. So this is the way that we used to analyze and ensure that this is real exchange in voluntary activation. So you can see these are trace forces. This is 20 responses. We repeat. We make a series of baselines to ensure that there is a change after the intervention. And I'm just showing you the change after the +2 intervention, and you can see that they were able to exert more force on average. And we see something very similar in the EMG responses. So force and EMG was potentiated. And finally, we asked the question, can this result in a functional relevant change? Not only in outcomes from EMG and force, but we test the 9-hole peg test to assess a hand motor function. And the individuals were instructed to pick any one of these pins as fast as possible and position this to the side, and then go back and reposition them in the same initial position. And we were able to observe that the speed of the movement, they were able to exert the motor task faster compared to the initial position. So we're currently doing 2 different series of experiments to follow up this result. One is we're using an NMDA receptor agonist to see if this is really the mechanism mediating this type of changes in excitability. And the second one is, can we potentiate this effect by using other possible interventions such as voluntary movement, or we're stimulating people for 2 weeks approximately and see if we can enhance the voluntary output. So I have shown you some evidence that spike time-dependent plasticity-like changes can be elicited at residual corticospinal synapses after human spinal cord injury. We propose that residual corticospinal synapses might represent a new avenue for enhancing the recovery of motor function and voluntary motor output. And I think what is very important, and I just want to finalize my talk with this message, is that a human electrophysiology can be quite valuable, and perhaps it can have a role in helping us to determine how can we recover better our patients with spinal cord injury. And I just want to thank the people in my group, Karen, Tochiki, John, Finn, and the students, particularly Karen Bundai, who was involved largely in those experiments in spike time-dependent plasticity, and all the funding agencies who allow us to do our work. Thank you very much. Yes.
Back to the last graph you had in there?
Sure.
It looked like there was, I didn't quite understand it, but there was something, a message you were trying to deliver with it.
Mm-hmm.
What's going on here? I mean, I see the vertical axis, but I don't understand quite what you're doing with the horizontal axis.
Yes, the idea is to show that we calculated the improvements in speed, the differences in speed, so after the intervention, they were able to do the movement faster. Okay, they were 15 or 20%, 15% faster than at the beginning of the test. So they spend less time.
Is this block in this rectangle?
Yes, the intervention will be the time. Yes, let me go back. Or any of the times, really. The intervention will be, we took the measurements before, and then this is the time, 17 minutes, where they received the pair stimulation. And after the intervention, we did a series of measurements in different sessions, either physiological measurements, or we also quantified voluntary output. Yes, so they were faster in performing the task, 10-15% faster compared to their baseline.
So when you're looking at the data point at 10 minutes there, nothing happened between 0 and 10, and then they started doing this exercise and moving the pins.
We never took physiological measurements and behavioral measurements in the same session. First, we characterize all the physiology, because the physiological measurements can be affected by just repetition of the task. Okay, so we were extremely careful in first characterize there is a physiological change, and if there is, do we see any changes in motor behavior? So we were able to reproduce the effect several times by patients coming back to the lab. So then we tested the motor output, then they did the intervention, and then we tested motor output. But we never mixed physiology. We try to tend to do that in separate sessions for effects. They can be contaminated, yeah.
You said they were able to perform tasks faster. Was there any time where enhancing the corticospinal transmission made a difference between being able to perform tasks? Like they could not perform before, but then after the enhancement of the transmission, they were able to perform.
Yes, that's it. We didn't look at that. We just look at very brief ballistic isometric tasks. So we didn't really explore if this change will result in being able to make another task, for example, or being able to make another movement that was not trained. So right now we're including kinematic analysis also. So we will be able to detect if there are small changes in the wrist to grasp movement also during this intervention. At that point was mainly looking at voluntary output and do you make the task faster. And we really target the 9-hole pec test because you are largely using the first dorsal interosseous muscle, which is the muscle that we target for the intervention. But it's a good point. We didn't really look at any other aspects of the task that could have been modified or not. One of the reviewers for this paper asked, did subjects have more or less phantom limb sensations after the intervention by the electrical stimulation in motor cortex? And we had 2 reports after the 18 people that reported that perhaps they noticed some difference, but there was more an anecdotal report than, it was not something that we could continuously see in all our patients, yeah.
The dexterity test you showed at the last, the PEG test, that improved. And so how is it possible? Because the stimulation paradigm that you're using is specific to certain kind of muscles, or at least dexterity is sort of broad across the large muscles.
Yes, that's what we use the 9-hole pec test primarily because for that task, primarily we restrict most of the arm movements and we ask them to do this type of movement, which is largely involving the first dorsal interosseous and the thumb muscles. And both 2 muscles are largely intermingled in the representation in the motor cortex. So it's more likely that at the intensities that we use, not only one intrinsic finger muscle has been affected, but we selected that task particularly because you don't have a gross movement that is involved in the task. We limit the movement as much as possible to try to detect a more specific change perhaps in a muscle that is involved in the task. And why do we think that they move faster? It's because they're able to exert more EMG. Okay, they're able to exert more force and more EMG activity, and we know that when you perform a task faster, you exert more EMG activity. So there is a correlation between the velocity of a movement and the magnitude of EMG. So that for us was perhaps one of the mechanisms that contribute to that you become faster. The intervention allows you to exert more EMG, therefore you become faster. Therefore, the 9-hole pec test perhaps you execute it in a different way after the intervention. Yeah.
Excellent work. Thank you very much for showing this. How did you decide on the number of stimulations that you provided? You said it was 17?
Yes.
17
we use 100 pair of pulses for 17 minutes. We started using 50 pair of pulses, which is what other groups have shown to be effective in motor cortex. And we observed that in patients, it was not enough. We did a pilot experiment at the beginning. So 100 pair of pulses the dose didn't seem to be effective. So we use 100 pair of pulses. We haven't done carefully a dose experiment. These are very long, prolonged experiments. So we decided to go from 50 to 100 pulses. It could be that if you use 200 pulses, it's more or less effective. We haven't tested that. But the rationale is coming from that. We started with 50, which others have used. The effect was more variable, so we increased the number of pulses, yes.
And do you, at what time do you see the effect wear off? Have you been able to follow
Yes,
that?
yes, that's a good question. The physiological data, we follow up subjects, and it went back to baseline at 80 minutes approximately. It didn't last longer. So you can see the evoked responses, the amplitude went back to the amplitude and the baseline around 80 minutes. Mm-hmm.
Monica, with all the activity going around about discomplete injuries, et cetera, these are studied. Your studies are all in Asia C and Asia D patients, right?
Yes.
So, with all this discussion going around about discomplete injuries, and the level of completeness even in Asia A's and all, and B's, it leads you to sort of think, well, what would happen if you did these studies in those patients? Have you tested them at all?
No, unfortunately, in order for us to make this estimation, we need to be able to elicit descending responses from motor cortex. We have been able, if we have a patient which is ACIA or have complete motor paralysis, we have been able, by increasing the stimulus intensity or using 2 pulses at the right interval, we have been able to see some residual connections. Okay, so a complete paralysis, the definition of clinical definition of complete versus perhaps a physiological definition of what is complete might not always go together. Yes, exactly. Yes. So we have tried. We had people that have asked us to examine this possibility, and it could be that the only thing that we need is to be able to determine a latency. So we need to be able to at least evoke some response to be able to know when do we need to stimulate. It doesn't have to be a large response. It could be a threshold response. But we need to have some descending, some remaining descending pathway, even if we stimulate electrically, okay, the corticospinal axons, to at least to be able to guide and determine where do we stimulate, when, not where, but when do we stimulate. Yeah, so that's a good question. It could be that more patients will benefit, or we could increase the number of patients. Unfortunately, the equipment that we have is limited also by the intensity. There was a group that was trying to develop a magnetic stimulator, put 3 together to see if you could have a more powerful output, because it could be that they have those fibers available, and we have seen that repeatedly. It's just not enough intensity to activate them. Okay, and you should see the intensity that we need to elicit responses from the area of the injury. It's high, it's very high, but we can do so. The definition of what is really complete becomes, yes, perhaps with this type of assessment, it will tell you that there is something still there. Yes, yeah, that's a good, very, very good point. Yeah. Yes.
So if the effect seems to dwindle after 80 minutes, for a future functional therapeutic thing, are you envisioning, enhancing the synaptic, strengthening the synaptic connections temporarily, and then applying other therapeutic techniques?
Yes, we're using 2 approaches. One of them, we're asking some of the same subjects to go back to the lab and do the same protocol on a daily basis. We still don't have enough data to support the view if this is really more effective or not. But we're using that approach to potentiate the effect. We're presenting some data at the Society for Neuroscience. We use the same protocol now during voluntary activity. So we ask the patients, so we change the threshold of motor neurons just by voluntary activating, and then we do the same stimulation, and it's more effective. We have data in 12 patients, and we will be showing that. So, but that's a single session. We don't know what happened repetitively. So but that's a good question, yeah.
The mechanisms of this plasticity is where?
The spinal cord.
Spinal cord.
Yes, yes. I mean, at least I think we show some evidence that is subcortical. We look at excitability of the motor neurons, and they didn't change. Okay, so that means it's not a purely change in motor neuronal excitability. It is a subcortical change. So most likely there has to be something happening at that level of the spinal cord, at the synaptic interaction. Yes. We think we don't know more what kind of receptors, and we just started to look at NMDA receptor, but we don't have enough data yet. Mm-hmm.
Is there an analogous experiment that you could do in the lower extremity?
Yeah, we are hoping to do this in the lower limb. Yes, yes.
How do you describe that experimental protocol?
Protocol. Okay, we will have to do exactly the same, look for the same type of latencies, but instead of looking for an intrinsic finger muscle, we will have to go to the tibialis anterior, for example. Yeah. Yes, yeah. I think there's different muscles that you could target, but the TA is largely cortically driven, the control of that muscle. So, and I think it will be, a good way to use the same technique in the lower limb muscle and see if you could potentiate the activity. Yes, absolutely.
We're going to have some fun talking about showing you some things that we're doing with incomplete,
good.
injuries that, maybe guide some of the selection of those muscles that you might choose to study. Yeah.
Mm-hmm. Okay. Yeah, excellent. I think this is in principle a phenomenon that was shown in animal experiments. We show the phenomenon in this particular muscle group, but as long as you can elicit responses and calculate latencies in a reliable way, you could do exactly the same for the lower limb. The hypothesis will be that, it potentially could have a similar effect, perhaps. Yeah.
In many of our patients, the function is quite variable from day to day. I was wondering if you follow an individual over time measuring the corticospinal excitability.
Yes, and that's one of the reasons why we use very simple task, a very, very simple task. We use, for example, for this protocol, the isometric voluntary ballistic task with the index finger. We didn't use a task that involved many muscle groups or many variables that could contribute to the task. So right now, to really understand if there was a change, at least we use a very simplistic task. The goal is, of course, to extrapolate this to a more general type of task. But as you well said, it's difficult to be able to measure times changes over time. But we're doing an experiment also where we have markers in both upper limbs, and we ask subjects to reach to grasp a very small object and also a very large object. Okay, and we're trying to quantify this variable first and see if there is some of those measurements that we can reliably use in order to measure performance. Because if we use the upper extremity motor scores, there's so gross motor tasks that it's very difficult to detect a change. We are hoping to be able to use more global tasks, but we're not there yet. Yes?
With the magnetic stimulator, you should be able to generate high-frequency bursts for short times, maybe up to 100 hertz, and then try to see if you can induce LTP in the spinal cord and have long-lasting changes in synaptic plasticity.
Mm-hmm.
You try that?
Yeah, others have tried. There are 3 papers published who try to use high-frequency RTMS over motor cortex. 2 of them used in the hand representation, and 1 of them in the leg representation of the motor cortex. They use 5 hertz, and they tried to change sensory and motor output. They did that repetitively over 5 sessions or 10 sessions, and they seem to show the results are very variable. Okay, they seem to show changes in the evoked response, but this is what actually we're arguing that perhaps it's not a local change in excitability. Perhaps in order to change behavior, we have to go to the interaction between the corticospinal and the motor neurons to change the recovery. Perhaps those local changes in others have shown that, and it could be beneficial. The results, if you look at the papers, they're controversial. Some measurements are improved, some others are not. Could be many factors that contribute to that, but our goal is to perhaps provide another target for inducing a similar type of plasticity. Yeah. Yes.
Separate it out though, just the pure corticospinal transmission based on looking at the percentage change in amplitude.
Can you
separate it out in your paradigm right now or in a follow-up where you can purely say, granted, I know you said the corticospinal excitability didn't change, the motor thresholds didn't change, but motor thresholds alone may not indicate corticospinal excitability. So have you looked at in your current data or is there follow-up planning to, is there a way to isolate just the corticospinal outburst that is purely related to the motor cortical neuron excitation from the plasticity of the spine?
But, well, I mean, I guess you could, yes. For example, let's say you quantify central conduction time and you quantify those I waves that we are trying to quantify in EMG electrodes. It's very difficult. I think the only way to just separate the upper part from the motor neurons will be see if there is a changes in central conduction time. You know, if the central conduction time is modified before and after your intervention, for example. It's difficult with TMS only activate one, you know, we have evidence that it's primarily the corticospinal tract, but there could be other pathways that also are activated. We activate monosynaptic, disynaptic pathways. So, but we have good evidence that indeed it is corticospinal responses, okay? But if you want to separate, I guess the only perhaps indication will be to look at central conduction time alone and see if that part changed. That doesn't tell you anything about transmission through the motor neurons, but at least to see if potentially there could be a change. Maybe that would be a way. Yeah.
I'd like to definitely thank Monica for this.
Oh, no, thank you so much.
Talking about appreciation.
Thank you.
I want to give a plug for next month's speaker, Dr. Maron Bixon. He's going to be talking about direct current stimulation in case we haven't gotten enough brain stimulation. The day of that talk. The 6th, December 6th. Okay. So hopefully we'll see you all there.
Thanks so much.